Structure. 3 point for the structure of proteins, primary. Structure. Purification of a protein is usually only a prelude to a detailed biochemical dissection of its structure and function. What is it that makes one protein an enzyme, another a hormone, another a structural protein, and still another an antibody? How do they differ chemically? The most obvious distinctions are structural, and to protein structure we now turn. Figure 3. The structure of large molecules such as proteins can be described at several levels of complexity, arranged in a kind of conceptual hierarchy. For levels of protein structure are commonly defined, Fig. 3-23. A description of all covalent bonds, mainly peptide bonds and disulfide bonds, linking amino acid residues in a polypeptide chain is its primary structure. The most important element of primary structure is the sequence of amino acid residues. Secondary structure refers to particularly stable arrangements of amino acid residues giving rise to recurring structural patterns. Tertiary structure describes all aspects of the three-dimensional folding of a polypeptide. When a protein has two or more polypeptide subunits, their arrangement in space is referred to as quaternary structure. Our exploration of proteins will eventually include complex protein machines consisting of dozens to thousands of subunits. Primary structure is the focus of the remainder of this chapter. The higher levels of structure are discussed in Chapter 4. Differences in primary structure can be especially informative. Each protein has a distinctive number and sequence of amino acid residues. As we shall see in Chapter 4, the primary structure of a protein determines how it folds up into its unique three-dimensional structure, and this in turn determines the function of the protein. We first consider empirical clues that amino acid sequence and protein function are closely linked, then describe how amino acid sequence is determined. Finally, we outline the many uses to which this information can be put. The function of a protein depends on its amino acid sequence. The bacterium Escherichia coli produces more than 3,000 different proteins. A human has 20,000 genes encoding a much larger number of proteins, through genetic processes discussed in Part 3 of this text. In both cases, each type of protein has a unique amino acid sequence that confers a particular three-dimensional structure. This structure in turn confers a unique function. Some simple observations illustrate the importance of primary structure, or the amino acid sequence of a protein. First, as we have already noted, proteins with different functions always have different amino acid sequences. Second, thousands of human genetic diseases have been traced to the production of defective proteins. The defect can range from a single change in the amino acid sequence, as in sickle cell disease, described in Chapter 5, to deletion of a larger portion of the polypeptide chain, as in most cases of Duchenne muscular dystrophy, a large deletion in the gene encoding the protein dystrophin leads to production of a shortened, inactive protein. Finally, on comparing functionally similar proteins from different species, we find that these proteins often have similar amino acid sequences. Thus, a close link between protein primary structure and function is evident. Is the amino acid sequence absolutely fixed, or invariant, for a particular protein? No, some flexibility is possible. An estimated 20% to 30% of the proteins in humans are polymorphic, having amino acid sequence variants in the human population. Many of these variations in sequence have little or no effect on the function of the protein. Furthermore, proteins that carry out a broadly similar function in distantly related species can differ greatly in overall size and amino acid sequence. Although the amino acid sequence in some regions of the primary structure might vary considerably without affecting biological function, most proteins contain crucial regions that are essential to their function, and thus have sequences that are conserved. The fraction of the overall sequence that is critical varies from protein to protein, complicating the task of relating sequence to three-dimensional structure and structure to function. Before we can consider this problem further, however, we must examine how sequence information is obtained. The amino acid sequences of millions of proteins have been determined. Two major discoveries in 1953 were of crucial importance in the history of biochemistry. In that year, James D. Watson and Francis Crick deduced the double helical structure of DNA and proposed a structural basis for its precise replication, Chapter 8. Their proposal illuminated the molecular reality behind the idea of a gene. In the same year, Frederick Sanger worked out the sequence of amino acid residues in the polypeptide chains of the hormone insulin, Fig. 3-24, surprising many researchers who had long thought that determining the amino acid sequence of a polypeptide would be a hopelessly difficult task. It quickly became evident that the nucleotide sequence in DNA and the amino acid sequence in proteins were somehow related. Barely a decade after these discoveries, the genetic code relating the nucleotide sequence of DNA to the amino acid sequence of protein molecules was elucidated, Chapter 27. The amino acid sequences of proteins are now most often derived indirectly from the DNA sequences in genome databases. However, an array of techniques derived from traditional methods of polypeptide sequencing still command an important place in protein chemistry. We will summarize the traditional method and mention a few of the techniques derived from it. Figure 3. Protein chemistry is enriched by methods derived from classical polypeptide sequencing. The methods used in the 1950s by Fred Sanger to determine the sequence of the protein insulin are summarized, in their modern form, in Figure 3-25. Few proteins are sequenced in this way now, at least in their entirety. As noted above, the sequence of a protein can usually be predicted from the sequence of the gene encoding it, information now readily available in ever-growing genomic databases. However, the traditional sequencing protocols have provided a rich array of tools for biochemists, and almost every step in Figure 3-25 makes use of methods that are widely used in biochemistry labs, sometimes in quite different contexts. Frederick Sanger. In the Figure 3-20 In the traditional scheme for sequencing large proteins, the amino-terminal amino acid residue was first labeled and its identity determined. The amino-terminal alpha-amino group can be labeled with 1-fluoro-2,4-dinitrobenzene, FDNB, dansyl chloride, or dabsyl chloride, Fig. 3-26. Figure 3-20. Figure 3. The chemical sequencing process itself is based on a two-step process developed by Per Edmund Fig. 3-27. The Edmund degradation procedure labels and removes only the amino-terminal residue from a peptide, leaving all other peptide bonds intact. The peptide is reacted with phenylisothiocyanate under mildly alkaline conditions, which converts the amino-terminal amino acid to a phenylthiocarbamoyl, PTC, adduct. The peptide bond next to the PTC adduct is then cleaved in a step carried out in anhydrous trifluoroacetic acid, with removal of the amino-terminal amino acid as an anilinothiozolinone derivative. The derivatized amino acid is extracted with organic solvents, converted to the more stable phenylthiohydantoin derivative by treatment with aqueous acid, and then identified. The use of sequential reactions carried out under first basic and then acidic conditions provides a means of controlling the entire process. Each reaction with the amino-terminal amino acid can go essentially to completion without affecting any of the other peptide bonds in the peptide. The process is repeated until, typically, as many as 40 sequential amino acid residues are identified. The reactions of the Edmund degradation have been automated. Figure 3. Form cis residues must be followed. To determine the sequence of large proteins, early developers of sequencing protocols had to devise methods to eliminate disulfide bonds and to cleave proteins precisely into smaller polypeptides. Two approaches to irreversible breakage of disulfide bonds are outlined in Figure 3-28. Enzymes called proteases catalyze the hydrolytic cleavage of peptide bonds. Some proteases cleave only the peptide bond adjacent to particular amino acid residues, Table 3-6, and thus fragment a polypeptide chain in a predictable and reproducible way. A few chemical reagents also cleave the peptide bond adjacent to specific residues. Among proteases, the digestive enzyme trypsin catalyzes the hydrolysis of only those peptide bonds in which the carbonyl group is contributed by either a Lys or an ARG residue, regardless of the length or amino acid sequence of the chain. A polypeptide with 3 Lys and or ARG residues will usually yield 4 smaller peptides on cleavage with trypsin. Moreover, all except one of these will have a carboxyl terminal Lys or ARG. The choice of a reagent to cleave the protein into smaller peptides can be aided by first determining the amino acid content of the entire protein, employing acid to reduce the protein to its constituent amino acids. Trypsin would be used only on proteins that have an appropriate number of Lys or ARG residues. Methods for polypeptide reagent, trypsin, submaximal chymotrypsin, staphylococcus, ASPM, pepsin, endoprotein, enzymogen, cyanogen, all In classical sequencing, a large protein would be cleaved into fragments twice, using a different protease or cleavage reagent each time so that the fragment endpoints differed. Both sets of fragments were purified and sequenced. The order in which the fragments appeared in the original protein could then be determined by examining the overlaps in sequence between the two sets of fragments. Even if no longer used to sequence entire proteins, the traditional sequencing methods are still valuable in the lab. The sequencing of some amino acids from the amino terminus using the Edmund chemistry is often sufficient to confirm the identity of a known protein that has just been purified, or to identify an unknown protein purified on the basis of an unusual activity. Techniques employed in individual steps of the traditional sequencing method are also useful for other purposes. For example, the methods used to break disulfide bonds can also be used to denature proteins when that is required. Furthermore, the effort to label the amino terminal amino acid residue led eventually to the development of an array of reagents that could react with specific groups on a protein. The same reagents used to label the amino terminal alpha amino group can be used to label the primary amines of Lys residues, Fig. 3-26. The sulfhydryl group on cis residues can be modified with iodoacetamides, malimides, benzyl halides, and bromomethyl ketones, Fig. 3-29. Other amino acid residues can be modified by reagents linked to a dye or other molecule to aid in protein detection or functional studies. Figure 3-20. Mass spectrometry offers an alternative method to determine amino acid sequences. Modern adaptations of mass spectrometry provide an important alternative to the sequencing methods described above. Mass spectrometry can provide a highly accurate measure of the molecular weight of a protein, but can also do much more. In particular, some variants of mass spectrometry can provide the sequences of multiple short polypeptide segments, 20 to 30 amino acid residues, in a protein sample quite rapidly. The mass spectrometer has long been an indispensable tool in chemistry. Molecules to be analyzed, referred to as analytes, are first ionized in a vacuum. When the newly charged molecules are introduced into an electric and/or magnetic field, their paths through the field are a function of their mass-to-charge ratio, m/z. This measured property of the ionized species can be used to deduce the mass, m, of the analyte with very high precision. Although mass spectrometry has been in use for many years, it could not be applied to macromolecules such as proteins and nucleic acids. The m/z measurements are made on molecules in the gas phase, and the heating or other treatment needed to transfer a macromolecule to the gas phase usually caused its rapid decomposition. In 1988, two different techniques were developed to overcome this problem. In one, proteins are placed in a light-absorbing matrix. With a short pulse of laser light, the proteins are ionized and then desorbed from the matrix into the vacuum system. This process, known as matrix-assisted laser desorption/ionization mass spectrometry, or MALDI-MS, has been successfully used to measure the mass of a wide range of macromolecules. In a second and equally successful method, macromolecules in solution are forced directly from the liquid to the gas phase. A solution of analytes is passed through a charged needle that is kept at a high electrical potential, dispersing the solution into a fine mist of charged microdroplets. The solvent surrounding the macromolecules rapidly evaporates, leaving multiply charged macromolecular ions in the gas phase. This technique is called Electrospray ionization mass spectrometry, or ESI-MS. Protons added during passage through the needle give additional charge to the macromolecule. The m/z of the molecule can be analyzed in the vacuum chamber. Mass spectrometry provides a wealth of information for proteomics research, enzymology, and protein chemistry in general. The techniques require only minuscule amounts of sample, so they can be readily applied to the small amounts of protein that can be extracted from a two-dimensional electrophoretic gel. The accurately measured molecular mass of a protein is critical to its identification. Once the mass of a protein is accurately known, mass spectrometry is a convenient and accurate method for detecting changes in mass due to the presence of bound cofactors, bound metal ions, covalent modifications, and so on. The process for determining the molecular mass of a protein with ESI-MS is illustrated in Figure 3-30. As it is injected into the gas phase, a protein acquires a variable number of protons, and thus positive charges, from the solvent. The variable addition of these charges creates a spectrum of species with different mass-to-charge ratios. Each successive peak corresponds to a species that differs from that of its neighboring peak by a charge difference of 1 and a mass difference of 1 proton. The mass of the protein can be determined from any two neighboring peaks. Figure 3-30. Source, information. Mass spectrometry can also be used to sequence short stretches of polypeptide, an application that has emerged as an invaluable tool for quickly identifying unknown proteins. Sequence information is extracted using a technique called tandem MS or MS/MS. A solution containing the protein under investigation is first treated with a protease or chemical reagent to hydrolyze it to a mixture of shorter peptides. The mixture is then injected into a device that is essentially two mass spectrometers in tandem, Fig. 331a top. In the first, the peptide mixture is sorted so that only one of the several types of peptides produced by cleavage emerges at the other end. The sample of the selected peptide, each molecule of which has a charge somewhere along its length, then travels through a vacuum chamber between the two mass spectrometers. In this collision cell, the peptide is further fragmented by high-energy impact with a collision gas such as helium or argon that is bled into the vacuum chamber. Each individual peptide is broken in only one place on average. Although the breaks are not hydrolytic, most occur at the peptide bonds. Figure 3. The second mass spectrometer then measures the m/z ratios of all the charged fragments. This process generates one or more sets of peaks. A given set of peaks, Fig. 331b, consists of all the charged fragments that were generated by breaking the same type of bond, but at different points in the peptide. One set of peaks includes only the fragments in which the charge was retained on the amino terminal side of the broken bonds, another includes only the fragments in which the charge was retained on the carboxyl terminal side of the broken bonds. Each successive peak in a given set has one less amino acid than the peak before. The difference in mass from peak to peak identifies the amino acid that was lost in each case, thus revealing the sequence of the peptide. The only ambiguities involve leucine and isoleucine, which have the same mass. Although multiple sets of peaks are usually generated, the two most prominent sets generally consist of charged fragments derived from breakage of the peptide bonds. The amino acid sequence derived from one set can be confirmed by the other, improving the confidence in the sequence information obtained. The various methods for obtaining protein sequence information complement one another. The Edmund Degradation procedure is sometimes convenient to get sequence information uniquely from the amino terminus of a protein or peptide. However, it is relatively slow and requires a larger sample than does mass spectrometry. Mass spectrometry can be used for small amounts of sample and for mixed samples. It provides sequence information, but the fragmentation processes can leave unpredictable sequence gaps. Although most protein sequences are now extracted from genomic DNA sequences, Chapter 8, by employing our understanding of the genetic code, Chapter 27, direct protein sequencing is often necessary to identify unknown protein samples. Both protein sequencing methods permit the unambiguous identification of newly purified proteins. Mass spectrometry is the method of choice to identify proteins that are present in small amounts. For example, the technique is sensitive enough to analyze the few hundred nanograms of protein that might be extracted from a single protein band on a polyacrylamide gel. Direct sequencing by mass spectrometry also can reveal the addition of phosphoryl groups or other modifications, Chapter 6. Sequencing by either method can reveal changes in protein sequence that result from the editing of messenger RNA in eukaryotes, Chapter 26. Thus, these methods are all part of a robust toolbox used to investigate proteins and their functions. Small peptides in proteins can be chemically synthesized. Many peptides are potentially useful as pharmacologic agents, and their production is of considerable commercial importance. There are three ways to obtain a peptide: 1. Purification from tissue, a task often made difficult by the vanishingly low concentrations of some peptides; 2. Genetic engineering, Chapter 9, or 3. Direct chemical synthesis. Powerful techniques now make direct chemical synthesis an attractive option in many cases. In addition to commercial applications, the synthesis of specific peptide portions of larger proteins is an increasingly important tool for the study of protein structure and function. The complexity of proteins makes the traditional synthetic approaches of organic chemistry impractical for peptides with more than 4 or 5 amino acid residues. One problem is the difficulty of purifying the product after each step. The major breakthrough in this technology was provided by R. Bruce Merrifield in 1962. His innovation was to synthesize a peptide while keeping it attached at one end to a solid support. The support is an insoluble polymer, resin, contained within a column, similar to that used for chromatographic procedures. The peptide is built up on this support one amino acid at a time, through a standard set of reactions in a repeating cycle, Fig. 3 to 32. At each successive step in the cycle, protective chemical groups block unwanted reactions. Figure 3 to 32 chemical. Red. R. Bruce Merrifield. The technology for chemical peptide synthesis is now automated. An important limitation of the process, a limitation shared by the Edmund Degradation sequencing process, is the efficiency of each chemical cycle, as can be seen by calculating the overall yields of peptides of various lengths when the yield for addition of each new amino acid is 96% versus 99.8%, Table 3 to 7. Incomplete reaction at one stage can lead to formation of an impurity, in the form of a shorter peptide, in the next. The chemistry has been optimized to permit the synthesis of proteins of 100 amino acid residues in a few days in reasonable yield. A very similar approach is used to synthesize nucleic acids, see Fig. 8 to 33. It is worth noting that this technology, impressive as it is, still pales when compared with biological processes. The same 100 residue protein would be synthesized with exquisite fidelity in about 5 seconds in a bacterial cell. Table 3. Yield in overall number of the final 11, 21, 31, 51, 100. A variety of new methods for the efficient ligation, joining together, of peptides has made possible the assembly of synthetic peptides into larger polypeptides and proteins. With these methods, novel forms of proteins can be created with precisely positioned chemical groups, including those that might not normally be found in a cellular protein. These novel forms provide new ways to test theories of enzyme catalysis, to create proteins with new chemical properties, and to design protein sequences that will fold into particular structures. This last application provides the ultimate test of our increasing ability to relate the primary structure of a peptide to the three-dimensional structure that it takes up in solution. Amino acid sequences provide important biochemical information. Knowledge of the sequence of amino acids in a protein can offer insights into its three-dimensional structure and its function, cellular location, and evolution. Most of these insights are derived by searching for similarities between a protein of interest and previously studied proteins. Thousands of sequences are known and available in databases accessible through the Internet. A comparison of a newly obtained sequence with this large bank of stored sequences often reveals relationships both surprising and enlightening. Exactly how the amino acid sequence determines three-dimensional structure is not understood in detail, nor can we always predict function from sequence. However, protein families that have some shared structural or functional features can be readily identified on the basis of amino acid sequence similarities. Individual proteins are assigned to families based on the degree of similarity in amino acid sequence. Members of a family are usually identical across 25% or more of their sequences and proteins in these families generally share at least some structural and functional characteristics. Some families, however, are defined by identities involving only a few amino acid residues that are critical to a certain function. A number of similar substructures, or domains, to be defined more fully in Chapter 4, occur in many functionally unrelated proteins. These domains often fold into structural configurations that have an unusual degree of stability, or that are specialized for a certain environment. Evolutionary relationships can also be inferred from the structural and functional similarities within protein families. Certain amino acid sequences serve as signals that determine the cellular location, chemical modification, and half-life of a protein. Special signal sequences, usually at the amino terminus, are used to target certain proteins for export from the cell. Other proteins are targeted for distribution to the nucleus, the cell surface, the cytosol, or other cellular locations. Other sequences act as attachment sites for prosthetic groups, such as sugar groups in glycoproteins and lipids in lipoproteins. Some of these signals are well characterized and are easily recognized in the sequence of a newly characterized protein, Chapter 27. Key convention. Much of the functional information encapsulated in protein sequences comes in the form of consensus sequences. This term is applied to such sequences in DNA, RNA, or protein. When a series of related nucleic acid or protein sequences are compared, a consensus sequence is the one that reflects the most common base or amino acid at each position. Parts of the sequence that have particularly good agreement often represent evolutionarily conserved functional domains. A range of mathematical tools available on the Internet can be used to generate consensus sequences or identify them in sequence databases. Box 3-2 illustrates common conventions for displaying consensus sequences. Protein sequences help elucidate the history of life on Earth. The simple string of letters denoting the amino acid sequence of a protein holds a surprising wealth of information. As more protein sequences have become available, the development of more powerful methods for extracting information from them has become a major biochemical enterprise. Analysis of the information available in the ever-expanding biological databases, including gene and protein sequences and macromolecular structures, has given rise to the new field of bioinformatics. One outcome of this discipline is a growing suite of computer programs, many readily available on the Internet, that can be used by any scientist, student, or knowledgeable layperson. Each protein's function relies on its three-dimensional structure, which in turn is determined largely by its primary structure. Thus, the biochemical information conveyed by a protein sequence is limited only by our understanding of structural and functional principles. The constantly evolving tools of bioinformatics make it possible to identify functional segments in new proteins and help establish both their sequence and their structural relationships to proteins already in the databases. On a different level of inquiry, protein sequences are beginning to tell us how the proteins evolved and, ultimately, how life evolved on this planet. Box 3. Consensus. Figure 1 representation. In one type, when multiple amino acids. The field of molecular evolution is often traced to Emil Zuckerkandl and Linus Pauling, whose work in the mid-1960s advanced the use of nucleotide and protein sequences to explore evolution. The premise is deceptively straightforward. If two organisms are closely related, the sequences of their genes and proteins should be similar. The sequences increasingly diverge as the evolutionary distance between two organisms increases. The promise of this approach began to be realized in the 1970s, when Carl Woese used ribosomal RNA sequences to define the archaea as a group of living organisms distinct from the bacteria and eukarya, see Fig. 1-5. Protein sequences offer an opportunity to greatly refine the available information. With the advent of genome projects investigating organisms from bacteria to humans, the number of available sequences is growing at an enormous rate. This information can be used to trace biological history. The challenge is in learning to read the genetic hieroglyphics. Evolution has not taken a simple linear path. Complexities abound in any attempt to mine the evolutionary information stored in protein sequences. For a given protein, the amino acid residues essential for the activity of the protein are conserved over evolutionary time. The residues that are less important to function may vary over time, that is, one amino acid may substitute for another, and these variable residues can provide the information to trace evolution. Amino acid substitutions are not always random, however. At some positions in the primary structure, the need to maintain protein function may mean that only particular amino acid substitutions can be tolerated. Some proteins have more variable amino acid residues than others. For these and other reasons, different proteins evolve at different rates. Another complicating factor in tracing evolutionary history is the rare transfer of a gene or group of genes from one organism to another, a process called horizontal gene transfer. The transferred genes may be similar to the genes they were derived from in the original organism, whereas most other genes in the same two organisms may be only distantly related. An example of horizontal gene transfer is the recent rapid spread of antibiotic resistance genes in bacterial populations. The proteins derived from these transferred genes would not be good candidates for the study of bacterial evolution because they share only a very limited evolutionary history with their host organisms. The study of molecular evolution generally focuses on families of closely related proteins. In most cases, the families chosen for analysis have essential functions in cellular metabolism that must have been present in the earliest viable cells, thus greatly reducing the chance that they were introduced relatively recently by horizontal gene transfer. For example, a protein called EF1-alpha, elongation factor 1-alpha, is involved in the synthesis of proteins in all eukaryotes. A similar protein, EF2, with the same function is found in bacteria. Similarities in sequence and function indicate that EF1-alpha and EF2 are members of a family of proteins that share a common ancestor. The members of protein families are called homologous proteins, or homologs. The concept of a homolog can be further refined. If two proteins in a family, that is, two homologs, are present in the same species, they are referred to as paralogs. Homologs from different species are called orthologs. The process of tracing evolution involves first identifying suitable families of homologous proteins, and then using them to reconstruct evolutionary paths. Homologs are identified through the use of increasingly powerful computer programs that can directly compare two or more chosen protein sequences, or can search vast databases to find the evolutionary relatives of one selected protein sequence. The electronic search process can be thought of as sliding one sequence past the other until a section with a good match is found. Within this sequence alignment, a positive score is assigned for each position where the amino acid residues in the two sequences are identical. The value of the score varying from one program to the next to provide a measure of the quality of the alignment. The process has some complications. Sometimes the proteins being compared match well at, say, two sequence segments, and these segments are connected by less related sequences of different lengths. Thus the two matching segments cannot be aligned at the same time. To handle this, the computer program introduces gaps in one of the sequences to bring the matching segments into register Fig. 3-33. Of course, if a sufficient number of gaps are introduced, almost any two sequences could be brought into some sort of alignment. To avoid uninformative alignments, the programs include penalties for each gap introduced, thus lowering the overall alignment score. With electronic trial and error, the program selects the alignment with the optimal score that maximizes identical amino acid residues while minimizing the introduction of gaps. Figure 3 to 30. Finding identical amino acids is often inadequate in attempts to identify related proteins or, more importantly, to determine how closely related the proteins are on an evolutionary timescale. A more useful analysis also considers the chemical properties of substituted amino acids. Many of the amino acid differences within a protein family may be conservative, that is, an amino acid residue is replaced by a residue having similar chemical properties. For example, a glue residue may substitute in one family member for the ASP residue found in another. Both amino acids are negatively charged. Such a conservative substitution should logically receive a higher score in a sequence alignment than does a non-conservative substitution, such as replacement of the ASP residue with a hydrophobic Phe residue. For most efforts to find homologies and explore evolutionary relationships, protein sequences, derived either directly from protein sequencing or from the sequencing of the DNA encoding the protein, are superior to non-genic nucleic acid sequences, those that do not encode a protein or functional RNA. For a nucleic acid, with its 4 different types of residues, random alignment of non-homologous sequences will generally yield matches for at least 25% of the positions. Introduction of a few gaps can often increase the fraction of matched residues to 40% or more, and the probability of chance alignment of unrelated sequences becomes quite high. The 20 different amino acid residues in proteins greatly lower the probability of uninformative chance alignments of this type. The programs used to generate a sequence alignment are complemented by methods that test the reliability of the alignments. A common computerized test is to shuffle the amino acid sequence of one of the proteins being compared to produce a random sequence, then to instruct the program to align the shuffled sequence with the other, unshuffled one. Scores are assigned to the new alignment, and the shuffling and alignment process is repeated many times. The original alignment, before shuffling, should have a score significantly higher than any of those within the distribution of scores generated by the random alignments. This increases the confidence that the sequence alignment has identified a pair of homologs. Note that the absence of a significant alignment score does not necessarily mean that no evolutionary relationship exists between two proteins, as we shall see in Chapter 4. Three-dimensional structural similarities sometimes reveal evolutionary relationships where sequence homology has been wiped away by time. To use a protein family to explore evolution, researchers identify family members with similar molecular functions in the widest possible range of organisms. Information from the family can then be used to trace the evolution of those organisms. By analyzing the sequence divergence in selected protein families, investigators can segregate organisms into classes based on their evolutionary relationships. This information must be reconciled with more classical examinations of the physiology and biochemistry of the organisms. Certain segments of a protein sequence may be found in the organisms of one taxonomic group, but not in other groups. These segments can be used as signature sequences for the group in which they are found. An example of a signature sequence is an insertion of 12 amino acids near the amino terminus of the EF1-alpha/EF2 proteins in all archaea and eukaryotes, but not in bacteria. Fig. 3-34. This particular signature is one of many biochemical clues that can help establish the evolutionary relatedness of eukaryotes and archaea. Signature sequences have been used to establish evolutionary relationships among groups of organisms at many different taxonomic levels. By considering the entire sequence of a protein, researchers can now construct more elaborate evolutionary trees with many species in each taxonomic group. Figure 3-35 presents one such tree for bacteria based on sequence divergence in the protein GRO-EL, a protein present in all bacteria that assists in the proper folding of proteins. The tree can be refined by basing it on the sequences of multiple proteins, and by supplementing the sequence information with data on the unique biochemical and physiological properties of each species. There are many methods for generating trees, each method with its own advantages and shortcomings, and many ways to represent the resulting evolutionary relationships. In Figure 3 to 35, the free endpoints of lines are called external nodes. Each represents an extant species, and each is so labeled. The points where two lines come together, the internal nodes, represent extinct ancestor species. In most representations, including Fig. 3 to 35, the lengths of the lines connecting the nodes are proportional to the number of amino acid substitutions separating one species from another. If we trace two extant species to a common internal node, representing the common ancestor of the two species, the length of the branch connecting each external node to the internal node represents the number of amino acid substitutions separating one extant species from this ancestor. The sum of the lengths of all the line segments that connect an extant species to another extant species through a common ancestor reflects the number of substitutions separating the two extant species. To determine how much time was needed for the various species to diverge, the tree must be calibrated by comparing it with information from the fossil record and other sources. Figure 3 to 34, a signature sequence in the EF1F2. Figure 3 to 35, evolutionary. As more sequence information is made available in databases, we can generate evolutionary trees based on multiple proteins. And we can refine these trees as additional genomic information emerges from increasingly sophisticated methods of analysis. All of this work moves us toward the goal of creating a detailed tree of life that describes the evolution and relationship of every organism on Earth. The story is a work in progress, of course. Fig. 3 to 36. The questions being asked and answered are fundamental to how humans view themselves and the world around them. The field of molecular evolution promises to be among the most vibrant of the scientific frontiers in the 21st century. Figure 3 to 30. Summary 3. For the structure of proteins. Primary structure. Differences in protein function result from differences in amino acid composition and sequence. Some variations in sequence may occur in a particular protein, with little or no effect on its function. Amino acid sequences are deduced by fragmenting polypeptides into smaller peptides with reagents known to cleave specific peptide bonds, determining the amino acid sequence of each fragment by the automated Edmund Degradation procedure, and then ordering the peptide fragments by finding sequence overlaps between fragments generated by different reagents. A protein sequence can also be deduced from the nucleotide sequence of its corresponding gene in DNA, or by mass spectrometry. Short proteins and peptides, up to about 100 residues, can be chemically synthesized. The peptide is built up, one amino acid residue at a time, while tethered to a solid support. Protein sequences are a rich source of information about protein structure and function, as well as the evolution of life on Earth. Sophisticated methods are being developed to trace evolution by analyzing the slow changes in amino acid sequences of homologous proteins.